All engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to the Naked Scientists podcast, the programme where we bring you the biggest breakthroughs and talk to the major movers and shakers in the worlds of science, technology and medicine. I'm Chris Smith, and in this episode we're going to revisit some of the most magical moments and scientific milestones that we enjoyed in 2025, including the incredible legacy of Dame Jane Goodall, the brainwave reading bionic knee, why Labradors are so greedy, and the beer that doesn't give you a hangover. But first, 2025 saw the birth of a baby girl called Amy, who made medical history as the first child in the UK to be delivered from a transplanted uterus. Amy's mother, Grace, was born with a condition meaning she had no uterus of her own, and therefore no prospect of a natural pregnancy. I spoke to Richard Smith and his colleague Isabel Quiroga about how they helped to bring Amy into the world.
This is a 25-year process whereby we were very happily delivered a little baby, Amy Isabel, and she came from Britain's first transplanted uterus, and the uterus had been donated by the recipient's sister called Amy. This is just fantastic, I have to say, for all of us. It's such a happy family and really great to see.
What was the reason for needing to do this, Richard?
All the people who are coming to us for potential transplants are people who either don't have a womb, and most of those people have been born without a womb. So in this case, the patient had MRKH, which is a syndrome whereby you're born with no womb.
But obviously no uterus means no prospect of a pregnancy, and that's the problem you're seeking to solve here.
Yeah, that's absolutely right. So up until now, the options for women were either to go down the route of surrogacy, which is country dependent, but it's also, there's quite a lot of issues with surrogacy. The other option, of course, is adoption. Well, now we have this third option.
Before we bring Isabel in, who is your partner in crime in this, talk us through what the process involves to get a uterus from one woman into another and then culminate in a pregnancy.
I suppose the easiest way is to go back 2 years ago to when we did that transplant. Two sisters were admitted, and there's the retrieval operation from Amy. It's a modified radical hysterectomy is the official term. So it's a long dissection, lasted about 8 hours, in fact. And then there's the implantation procedure. That's led by Isabel. Isabel does the vessel hookup, and then I do the kind of genital tract anatomical hookup with the vagina and the ligaments, etc.
Do you have the two patients in the same operating theatre, or are the two of you operating in adjacent theatres, Isabel? Where do you stand?
Yeah.
When does this get handed on to you?
We have two separate theatres. Due to infection control, you can't have two patients in a same operating room. So the way it happens, where we start the donor operation, when we are at a point in which the organ comes out, then it comes to a small operating room, side table, in which we can prepare the organ for implantation. With any organ, the most important thing to do once it comes out is to remove the blood. We don't want little clots within the vessel in the organ, so we flush all the blood out. Once that we have the organ ready for transplant, then we can go with the organ to the other theatre, adjacent theatre. The recipient would have already been anaesthetized. The operation, getting the vessels ready, which are the vessels that are going to go and provide the blood supply, have already been made for us to join the uterus vessels onto, so the organ gets its blood supply from the recipient.
And do you see the thing, as soon as you take the clamps off, do you see the blood flowing and it go a beautiful red colour so you know you're in business? Is that how you know, right, we've got a good chance of this working?
Absolutely. So all organs, when they're cold and when they're deprived of blood, have a pale colour. And so when the blood returns into the organ, you can see them changing colour. And actually, when you do kidney transplants, that's absolutely amazing. It's one of the wonders of what we do. So it's very similar in the uterus, and you see it changing colour and almost coming to life.
One of the things that was occurring to me, though, you are a transplant surgeon and you don't normally do uteruses. So how on earth did you get involved in this? And was it a steep learning curve to start doing uteruses?
Well, the story of how I got involved is a long one, but basically, Richard and his team were interested in doing transplant from deceased donors. And I lead one of the retrieval centres in the UK. They just asked for some advice how to get into the world of retrieval. And it just developed into this true partnership. I have to say that I was initially quite reluctant. I couldn't see the need for this operation. I kept worrying about the risks. But it was only when I met the first patient and I heard the pain and what she had gone through that I understood the need for this operation. And obviously I have changed my mind completely, and I'm a true believer in that this is a very worthwhile cause.
You must be delighted to hear that, Richard. You got a fan. But you come in and do the final sort of close up and do the genital anatomy to make sure that's correct. Having got the organ in situ now, and it looks like things are going well, talk us through what happens in the days, weeks, months after that.
Okay, so there's really close monitoring going on. We're doing cervical biopsies to look for rejection on a weekly basis at the start. We're doing Doppler flow ultrasound studies to look at blood flow within the muscle of the uterus. That's called the myometrium.
And how long do you allow before you're comfortable to say, right, I think we're ready to try to get a baby in here now?
So the minimum for that is 6 months, but there's various other factors that are brought into that. And certainly in this first case, we actually ran that process out to just over a year.
When you decide that the time is right and it's safe to initiate a pregnancy, where do the embryos that you're going to implant, where do they come from?
So they've all been created pre-transplant, and they're all in cold storage. And depending on age, they've all been pre-implantation genetic tested, meaning that we know that those embryos are genetically normal. And that's one of the conditions to being able to come into the trial.
And how many embryos do you put in?
Oh, one at a time only.
You go one at a time. And then how is the baby actually delivered? How do you decide when to deliver the baby and how?
So the pregnancy is monitored as per any other pregnancy, but there's a big issue for these women with a transplanted uterus that the uterus itself has no nerve supply so that they can't feel contractions. So obviously we were monitoring that very closely. The delivery itself is performed by caesarean section. Baby was delivered really beautifully, I have to say, and the baby cried within seconds of emerging into this world, which obviously for obstetricians is a marvellous noise, that's for sure, and for mum and dad, even more marvellous.
Indeed. And at that point, does the uterus stay in and can you have more babies, or does it have to come out?
No, so you've got the women have got a choice at this point. They can either have a completion hysterectomy 6 months post-delivery. We're certainly not in the business of removing the uterus at the time of caesarean section. Or you can go on and have one more baby, and once you've had one more, you can then have your completion hysterectomy 6 months after that. And the purpose of that is to restrict the time the uterus is in to a maximum of 5 years.
Were you both there for the delivery? Or did you only get to see this? So you actually were there when the baby came out?
We were assisting at the delivery.
So you're like surrogate parents almost in this. Must have been an amazing moment, though, to see you, because that's 25 years of hard graft for you to get to this point.
Yeah, there were a lot of tears shed, actually. I mean, I'm not often rendered speechless, but that day I was really very, very moving.
Bet you never thought this was how your career was going to go, Isabel, when you thought, I'm going to be a kidney transplant surgeon, end up making babies.
Yes, it's a totally a true new experience. As you say, I'm not often in a delivery room or seeing babies being born, so it was truly something very, very different and wonderful, actually.
A lovely story. Richard Smith and Isabel Quiroga. On the 1st of October, news spread around the globe that the world-renowned primatologist and conservationist Dame Jane Goodall had died. She was 91. Will Tingle and primatologist Catherine Hobater at the University of St Andrews looked back on her remarkable life.
I had two dreams. One dream I wanted to work among, study, live among, learn from animals. And as I grew older and read more and more books, I realized that most of all I wanted to go to Africa and work among and learn about African animals. And my second dream, I wanted to write books about them. So I feel that I've been incredibly fortunate to stand here and look back over my life and realize that to a large extent my childhood dreams have already been fulfilled.
Jane Goodall was born in London in 1934. From a very young age, she showed a fierce curiosity about the natural world and would spend hours in her garden and nearby parks, notebook in hand, carefully observing the animals around her. As a child, she dreamed of distant lands and exotic animals, sometimes pretending to be Tarzan swinging through the trees. Later on, she would joke that Tarzan had married the wrong Jane. Jane's passion for animals stayed with her through her adolescence. She famously taught herself to read the naturalist books she borrowed from the library and was determined one day to travel to Africa. Her family encouraged her curiosity, even though such ambitions were unusual for a young woman at the time. She went on to study at the University of Cambridge in the 1950s, earning a PhD in ethology, a rare accomplishment for someone with no formal university science background. Her early work laid the foundation for her fascination with chimpanzees, the creatures that would define her work.
In chimp society, and this is quite rare in a lot of the animal societies, any male, whether he's high-ranking, middle-ranking, or low-ranking, even if he has a disability, he is able to sire an infant. And I think it is because of this ability of all the males with all their varied genes to sire infants that we find so much individuality. I mean, one chimp is as different from another chimp as one person is from another person.
In 1960, Jane travelled to Tanzania to begin her landmark study of chimpanzees at Gombe Stream National Park. She was given the unprecedented opportunity to observe the chimps in their natural environment. Over decades, she revealed intelligence, emotion, and social complexities like tool use, which were behaviours previously believed to be solely human. Her work challenged long-held scientific assumptions and inspired millions around the world. Catherine Hobater, a primatologist at the University of St Andrews, recalls how profoundly Jane inspired her own career.
What Jane Goodall meant to me was this idea that you could be a scientist, and a good scientist at that, without necessarily having to become this idea of a sort of cold, objective observer of other individuals. That actually, if you cared about the individuals that you were spending time with, if you got to know them, if you got to know them as having personalities, that actually meant that you as a scientist could ask deeper, richer, more interesting questions about their world. And it was part of the goal of her mentor, Louis Leakey, when he sent her out there, was to send out somebody with a fresh perspective, to send out somebody who didn't have formal academic scientific training, because you would potentially be going in with an open mind. And it's not to say, you have to be an incredibly good observer to see the details of behavior. You have to just have patience by the bucket load. And that doesn't necessarily have to do with thinking about hypotheses and predictions and scientific training. And actually, she herself also talked about the fact that lack of a formal scientific background really paid off in terms of her ability to see with a fresh perspective. And then she had just that incredible ability to share and connect everybody else to what she saw.
Beyond her research, Jane founded the Jane Goodall Institute in 1977. Dedicated to protecting endangered species and promoting environmental education worldwide. Jane Goodall's life was one of adventure and discovery. She endured the difficulties of living in remote African forests in order to study chimpanzees. Through persistence and curiosity, she became a leading figure in primatology and a powerful voice for conservation. From her childhood in London to her fieldwork in Tanzania, her story shows how determination can help reshape our understanding of the natural world.
If she was here and she was around listening to all of this today, she would be the first person to say, don't be sad. What are you all doing talking about me and my legacy? There is a million things out there to do, and we just really need to be cracking on with them to make sure that we're protecting chimpanzees. And I think that sense of urgency, that sense of the fact that every single person can do something, even if it's small, everything adds up, that sense of purpose that she was able to share with that positive message. That's the inspirational legacy she leaves behind, and that's what she would want us all to be doing, would be to make sure that we are not the last generation who will be having the privileges she did to live alongside wild chimpanzees.
And she was an inspiration to me. She played no small part in my decision to pursue a career in zoology and conservation. And one quote of hers that continues to resonate with me is, "When you study animals in the wild, you realise how they live and understand the interconnection of everything and how every little species has a role to play. So you understand that everything is interconnected on this planet. And I believe to save everything else would be to save ourselves too." Thank you, Jane.
Will Tingle reflecting on the life of Jane Goodall, who died on the 1st of October 2025. She was 91. In the summer of 2025, researchers at MIT announced the development of a new bionic knee prosthesis. The device is directly integrated with the amputee's bone and muscle, allowing the brain and the prosthesis to work together so the user can move much more comfortably and naturally. We talked to the appropriately named Tony Hsu at MIT to find out how it works.
The prostheses that most people have, the ones that you can buy on the market, they help you walk, they help you go downstairs maybe, but they don't have any motors, and so they can't move like human legs do. So in our work, what we did was we attempted to more directly integrate the prosthesis with the human so that not only can you exchange energy with the device, but you can actually exchange neural information to inform the device how to move directly.
Is that purely motor, as in movements, Tony, or are you saying you can get some feedback, some sensation from the prosthesis and feed that back into the person, so they've also got some awareness of where in space their prosthetic is and how much force they're putting through it?
So the really cool part is that the way we approach the problem, where we integrate the prosthesis directly with the residual bone after the amputation, the person who has this prosthesis actually gets direct force feedback from that bone. Just like our skeleton is supposed to be directly loaded, we take that same approach and we take advantage of all the rich sensors that we have in our bones and our tissues to actually provide that person sensation of the prosthesis. So even a very moderate tap all the way at the prosthetic foot can be felt at the bone just because of the way that we've designed the mechanical interface.
What about that movement ability as well? Because that's the thing, as you say, when a person loses a body part, they lose the ability to move everything downstream of wherever they've had the amputation. So what have you done to try and address that?
The technique that we used in this paradigm is called the agonist-antagonist myoneural interface, which essentially takes muscles which used to be connected together and reconnects them after the amputation. So in this study, we provided people muscles that work together again, just in this case for the knee. But we take these muscles that already exist, that used to control the knee, and then we reattach them in a way that makes sense to the brain, that provides the person a sense of their phantom knee moving around in free space.
So the person thinks they're going to flex their knee. This would normally extend or stretch the muscle over their thigh and shorten the muscle round the back. You can sense the activity in the muscles that would want to do that, work out therefore what the joint would do, and make the joint behave in an appropriate way. So when it does move, it moves in a way the brain's almost expecting it to.
I think that's actually a perfect explanation. And then we call this the feed-forward direction, as in the signal comes from the brain and then the joint reproduces the movement that the signal contains. And then in the feedback direction, just like you mentioned earlier, we get sensation from the prosthesis, all these forces and impacts and movements that are conveyed through the bone back to the brain. And so in this way, we have the feedback direction and we close what we call the control loop.
And how does that work functionally? What do the patients or the wearers say about this compared to devices that are much more dumb, for want of a better phrase? They're just a passive prosthetic that they're strapped on.
Right. It's like, it's night and day. working with these patients in the lab, when they come in and first start using our experimental device, it's really an emotional moment, especially because it's been so long for some of them since they've had movement of this joint. So just even the ability to flex and extend a knee, just those two movements, right? Not doing anything fancy, not juggling balls or trying to avoid obstacles, just extending and flexing the knee, that can produce a really profound emotional impact on our patients.
So people get to grips with it quite quickly. It's not a steep learning curve for them. They can actually master this and get to grips with it fairly fast.
As scientists, I would say if we do our job correctly, then it's completely intuitive. The interface is invisible or seamless, and the person just uses the motor skills which they developed since they were 1 or 2 years old, right? To move their residual muscles, to move their phantom joints. And then if we do our job correctly, we can immediately reflect these at the knee.
Given that it sounds from what you're saying like people took to this like a duck to water and really, really genuinely appreciated it, is this likely to become the gold standard then for above knee amputations? This is what we would strive to offer patients from now on. Are you going in that direction?
This is definitely the school of thought that we are a part of, we as in the researchers on the paper. There are a couple of perspectives. One is that if you make the device smart enough on its own, like let's say use AI or really advanced math, if you make the device smart enough on its own, then it can do everything that the human would want it to do. But the other perspective is that there are just some movements that you can't get the device to do, especially very fast and dynamic movements. Think again about playing football, perhaps, or dodging obstacles as you're running through a forest. There are some really dynamic movements that perhaps require human intelligence. And so we've taken this latter approach where we put the human in control. We put the pilot in the seat, right? And we really think that if you just provide a means for the person with amputation to express themselves physically, physiologically, then not only will they get a better movement out of the device, but they actually feel more incorporated with the device. And that's also extremely important for psychological well-being.
Tony Hsu at MIT. Now, for over 2 decades, snake fanatic Tim Friede voluntarily injected himself with venoms from lethal snakes and willingly exposed himself to hundreds of bites. He was on a mission to save lives, and it was revealed in May of this year that antibodies found in his blood can actually protect against fatal envenomations from 13 of the world's deadliest snakes, including even cobras and mambas. Now, a pharmaceutical company has used the genetic code from Tim's immune cells to copy the recipes for the antibodies he makes to help them to develop a universal snakebite antivenom.
I've been into snakes since the age of 5, and that was actually my first bite by a snake. Not really that venomous, rear fang snake, and it scared me. But then I became interested in it. And ever since then, it was going to museums or going to zoos or going out with my buddies Herpin in high school. Not for anything venomous, because we only have 2 venomous snakes in Wisconsin. So we're basically just chasing colubrids. We're chasing garter snakes and milk snakes, things like that. And then I got into the venomous snakes. I got into cobras.
Tim began buying up these snakes from sources in Florida. He built a special lab in the basement of his home to house them. How many, though? I inquired.
At the max, I had 60.
60? And
Yeah.
what sorts of snakes?
Cobras, mambas.
Just the ones that are responsible for some of the most lethal venoms that we know of. Yeah. Was that what fascinated you? Was it the fact that you're dealing with some of the most dangerous snakes, or was there another motivation for being interested in those in particular?
Russell's viper, saw-scale viper, fer-de-lance, those are the big killers because they live next to people. They just couldn't find them. So I chose the next best thing, which were cobras, which kill a lot of people, and mambas. For me, mambas was my major goal. Half my bites alone, over 100 bites, are just from mambas. They're just so dangerous.
Indeed, about 20 years ago, Tim embarked on a mission to build his own immunity to help solve the snakebite problem around the world. Historically, we've made antivenom by injecting small quantities of venoms into animals like horses and then purifying the antibodies they produce. But it's not perfect. It's expensive, it's time-consuming, and the antibodies are not human, which can in and of itself produce problems. So how did Tim approach the problem to build up his immunity? And how did he administer this incredibly potent venom?
I was accidentally bitten by some cobras when I was milking them, but I was also at the same time injecting venom, so I had immunity.
But why were you injecting the venom?
To build immunity so I don't die.
Did you just do that off your own back then? So you milked the snakes, got the venom, and then you were just shooting up with it?
Yep.
Didn't you think, well, I might die?
Yeah, and I almost did die a couple times.
What, from injecting it?
September 12, 2001, I had 2 cobra bites back to back in an hour and flatlined. Ended up in a coma in ICU.
But when you injected the venom, how did you know how much to inject? And what on earth was it like when you put it in? Was it painful?
It's very painful. It's kind of like a bee sting, but way worse than that because like bees will yield 1 or 2 milligrams of venom, and venomous snakes can go 400 to 500 milligrams of venom.
Were you using all of the different snake venoms, or were you just focusing on one of your particular snakes at the time?
Yeah, no, not all. Out of the 600, no, not all. I focused on just individual snakes because that way, instead of mixing venom, I know what individual snakes feel like when I'm bitten by them or I inject venom because all snake venom is different.
And it was that very long-term, repeated exposure that's led to Tim's immune system producing a very powerful, very refined, but also very general response to the venom. He now has broadly neutralising antibodies that can bind to many different venoms and block them. But how does Tim's family feel about what he does? And why does his work actually matter?
They look at it like it's the coolest thing in the world that I'm all over TV or all over the press based on what I do. And they realize that it's important for humanity, for the people that do die from snake bite, that I represent them, even though I'm 8,000 miles away. So that's the way I looked at it, is I'm representing people I'm never going to meet.
So your immune response has enabled the discovery of the genetic code for the antibodies that can neutralise a lot of these venoms now, which is a massive step forward towards a universal venom antidote, isn't it? What will you do next then? Because as I understand it, of the 19 that were tested, you were able to help through what you've done with 13 of those 19, and that leaves a gap of some 6. So is this a question of exposing you to more snakes, or are they going to use a similar approach to then get the final 6 but elsewhere or from animal studies or something? What's the next step?
Yeah, they just have to pan out my antibodies and they have to figure out the ones that we need that we don't have right now.
So you are still the source. It's still going to come from you, the solution.
Yeah. I still give blood. That's my main job, is to give them blood.
That is such an extraordinary story. That was the snake fanatic eager to find a universal antivenom, Tim Freedy.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
This is the Naked Scientist podcast with me, Chris Smith, and today we are recapping the outstanding science stories of 2025. Still to come, what makes our pet Labradors so greedy, and a new boozeless beer that you can try out in dry January. But first, we're going underground. No, not to the 1980s hit for the Jam, but instead onto the real London Underground to find out how quantum mechanics is being used to keep track of the oldest and one of the most extensive subway systems in the world. This story was first broadcast in February 2025. My guides were TfL, Transport for London, engineers Steve Foote and Steve Venables, and Imperial College London physicist Joe Cotter.
So the London Underground network's really complicated and extensive. We've got 11 lines, there's 272 stations. We move approximately 1.2 billion people a year. We have 400 kilometres of track, and then we've got 5 different signalling systems. So it's a really complicated system to operate.
I've joked in the past, but it's actually true. You're moving more people under the streets of London every day than the populations of whole countries, like Sweden, for example.
I am still amazed at how complicated the system is and what we achieve each day, yes.
So tell us about the problem that, as you see it, that having better positioning of trains could help to surmount.
In terms of improving the way in which we maintain the asset and become more efficient and effective in the way that we do that, then having data is fundamental. Obviously modern technology would allow us to use the train as a sensor, and it could run down the tracks, collect data around all those assets. And then if we've got an accurate positioning of where that train has collected that data from, if there's an issue, we know exactly where to go back to collect that data. And if we keep running the trains and keep collecting the data, you can then start trending the changing condition over time. But you can only do that if you know that you're capturing the data in the same place each time.
I was talking to someone the other day who's working with car manufacturers. They're collecting data collected by cars as they drive across the country's roads. They're kind of regarding cars now as a sensor on wheels. They know where the car is because they have GPS. In a tunnel, that presumably does not work.
That's correct. GPS, you can't rely on it in the underground sections of our network, and therefore an alternative form of accurate positioning is needed. The technology that signalling uses identifies the section of railway that the train's operating on, but it doesn't give you the absolute position. So if you want to go and identify where there's a defect on the railway, you need the absolute position to know where to go to.
Got it. So you know a train is on a section of track between two signals, but if it has encountered a bump in the road, you don't know where that bump is except it's somewhere between A and B. Whereas if you had a really precise way of keeping tabs on where the train is at any moment in time, you could say, that's where the problem is, that's where we've got to send the repair guys to.
Yes, so the signalling system uses a train being in a geographic section in order to keep the trains safe, keep them separated. We do also have other infrastructure installed on the network that it doesn't give you that accuracy we desire. So using quantum sensor could give us the level of accuracy such that we could do, similar as you described with the cars, use a train as a sensor to look at lots of different assets.
Steve Foote. And this is where Dr. Joe Cotter, a physicist at Imperial College London, comes in. He's pioneering quantum sensors based on clouds of supercooled atoms, the movements of which he can read with exquisite precision and accuracy using a laser. And by adding up all the movements the atoms make, you can work out how far and how fast the thing holding them must have moved. In this case, that would be a train, but it could equally be a boat, a car, or a plane. In other words, this would give us another way of keeping tabs on position, but without relying solely on mechanisms like GPS, which can sometimes fail or even be misleading.
In my labs, what we're doing is developing a new kind of sensor that harnesses quantum mechanics to make more accurate measurement devices. One of the nice things about this approach, so inertial navigation, is that the ability to position yourself is self-contained in the vehicle that you're travelling in. So you don't rely on anybody outside. It's all on that vehicle.
In basic terms then, what is inertial positioning and how does that actually work? Before we get into the quantum way of doing it, what's the basic principle?
So inertial navigation relies on measuring the motion of the vehicle that you're in. So in particular with the acceleration or the rotation of the platform. And then you have to do some maths to convert those measurements of inertial signals into a change in position in a map frame. So for the London Underground along the track, for example.
So if I reach in my pocket, take out my phone, it knows that it's moved upwards, sideways, along a bit, at what sort of rate, over what sort of time, and it could then work out, working backwards, doing that maths you mentioned, where it must now be relative to where it started. Is that what you mean?
That's exactly right, yeah.
And you're saying you want to do that with atoms, quantum level, tiny particles. Why do we need to go any further and use atoms?
By harnessing quantum mechanics, we think we can make more accurate sensors that could enable you to navigate for longer in the future. So it's about a next generation kind of approach to this inertial navigation.
How does it work?
So for our quantum inertial sensors, we start by laser cooling a cloud of rubidium atoms to a few microkelvin in temperature, about a millionth of a degree above absolute zero.
And why do they have to be that cold?
When they're cold like that, their quantum properties start to come to the forefront. And so to describe their motion, we need to treat them like waves. And it's that wave-like nature that we take advantage of in our sensors.
How does that work then? We've got a sensor, it's got these very cold rubidium atoms. How do they know where they're moving? And how do you log that? How do you extract that information?
We have a laser and we release our atoms and the atoms interact with the laser. The laser is essentially just a wiggling electric field and we use those wiggles like a ruler. And so if it's fixed to the vehicle and our atoms are in free fall, we just measure how many of those wiggles in the laser the atom moves through in a given amount of time.
Put simply then, if the atoms are in one position and the train moves forward, the atoms are left behind a bit, so they're going to move down the ruler a bit, your laser ruler, and you can register that, and that would correspond to an acceleration in a certain direction. Is that how it's working?
That's exactly right. Yeah, that's the principle behind it. We've taken demonstrator systems on the tube already, and we've had some success.
What you took, basically, I've seen your gear in the lab here. It's a whole room. So how on earth did you get that on the underground?
So we've packaged up one of our sensors and deployed it on a London Underground train.
Did you get any funny looks? Other passengers look a bit strangely at you.
It was a test train. There were no passengers.
Okay. Can it work in 3 dimensions? You've mentioned a sort of ruler analogy. That's one axis. Can you do this in multiple axes so that you've literally got a tracking in 3-dimensional space system? Because that's going to be needed. If we're going to need to know where an aeroplane is or a boat or something, we're going to need all of the degrees of movement to be tracked.
Yes, absolutely. So in the laboratory, we already do it in 3 dimensions. For the transportable work we're doing with TfL, we're focusing on just one axis for now, just to overcome these engineering challenges and environmental challenges. But no, you're absolutely right. The next step will be to develop a new 6-axis sensor for the railways.
Joe Cotter, another London Underground engineer who was actually instrumental in getting this collaboration off the ground, is TfL's Steve Venables. I asked him, if they can get this to work, what sort of a difference it might make.
This has potential to change the landscape of how we operate our railway. If the railway breaks for some reason, we'd be able to fix it quicker, cheaper and more efficiently. And we'd also be able to understand how that asset's performing so we can fix it before it does fail.
I also like the juxtaposition of one of the world's biggest and oldest railway systems lining up with some of the world's newest cutting-edge navigation technology.
Disruptive technology like this has to be looked at. We can't continue doing the same things and expect different results. We need to start challenging how we look at things going forward. And this is just one example of where we're looking to partner with industry, academia, to develop our own internal capabilities.
TfL engineers Steve Foote and Steve Venables and Joe Cotter at Imperial College London. That piece was made in partnership with UK Research and Innovation. Next, to a story from March 2025, when I took my hungry Labradors to meet the canine aficionado Eleanor Raffan. Eleanor has carried out lots of research on whether genes carried by this particular breed of dogs, and their owners actually, oh dear for me, can make them particularly greedy.
Morning, Eleanor. How are you? 3 words on the letterbox. That's brilliant.
Come in.
Hello. I'm sorry.
Welcome to the madhouse. Yeah. Come through this in here. The last time we met, you were introducing me to dogs that eat too much because of genetics. But what really is the overriding question this time?
Well, I'm still interested in overweight dogs, but we are ultimately interested in how our genes can control why some people overeat and gain weight. And if we do that via the medium of dogs, then it tells us something about veterinary medicine too.
So dogs work the same way we do?
Very similarly, they're another large mammal with very similar genetics, actually.
How did you approach this then?
Well, we've done what's called a genome-wide association study. These days we can test markers that kind of are anchor points all the way along the genome, and we can test at each of those points whether one or other version of the genome is associated with obesity. And when we did that, it allows us to map where on our genetic code there is something associated with a trait, and in our case, to map down to particular genes which were associated with obesity in the Labrador population that we were looking at.
So you look at a dog and you ask, this dog's fatter, this dog's thinner. Are there any differences in these different parts of the genome? Because if there are differences, genes in that region could be responsible.
That's a beautiful explanation.
We've got Labradors here. They're making noise and they're definitely greedy. Does that actually help then? Is that why you went for Labradors? Because they're notorious for tending to put on weight?
Yeah, it certainly meant that we thought we might find something interesting because they've got this reputation for being complete chowhounds.
So how did you do the study? I mean, was this just domestic dogs, people like me, who would say, well, come and look at mine?
Yeah, exactly. We've got entirely pet dogs in our discovery study, and we took slobber samples from them, spongy swabs, mopping up a little bit of saliva, and you can get DNA. The thing that we were studying genetically was their tendency to put on weight, so we just saw how overweight they were. There are ways to put numbers on how fat a dog are. We use something called a body condition score. And then we put a number on how greedy they are, and actually on how...
How did you measure that?
We developed a questionnaire a few years ago, and it's quite simple, actually. We've got 35 statements, things like, my dog will eat anything, my dog is fussy about food. And so we can put a number not only on how food motivated is the more polite way of putting it, dogs are, but also on the extent to which owners control diet and exercise. So what we did was the mapping study to test the genes was with the condition score. So first we said what genes make a dog more prone to being overweight. And then when we could put a number on that and combine all of them together as a risk score for obesity, then we said, well, if you're a high-risk dog, why are you high risk? And we could use our greed score and see that actually the high-risk dogs were at risk because they were more foodie. They were the kind of dogs who pester you for food at the table or always snaffle a scrap when they're out and about. They're the ones who will patiently wait even for something like a carrot when you're chopping vegetables. And they're the ones who are really just forever persisting in their pursuit of food.
Knowing a shopping list of genes that might be linked to overweightness is one thing, but actually the mechanism of how those genes translate into that's the key thing, isn't it? Because that tells us where the interventions might be. So can you see possible ways in which these genes that you have linked to this behaviour, being overweight, are translating into that occurring?
Yeah, absolutely. What we were really struck by was that our top 5 genes also have links to human obesity. And our very top gene was called DEN-D1B, and that's the one that we pursued with studies in the lab. And we found that it acts as a dimmer switch to turn up or turn down a brain pathway, which is quite well studied, actually, about how the body regulates body weight. And by having probably slightly more of it or slightly less of it, it slightly turns up or down your hunger signalling.
And that's true in humans as well.
Yeah, the effect we got in dogs was quite big. If you are a dog that carries this risk variant, then you're about 8% fatter than others that don't. The effect in humans is really minute, and they could only find it in these huge populations. But the fact that there is shared biology across the species meant that it was worth pursuing.
And is it not also the point that if you have just a small imbalance in energy terms, in what you need, in terms of what you're actually eating compared to what you really need, because we live a long time, you've got plenty of time to slowly accumulate weight. So even if it's a small and subtle effect, it's still going to translate into quite a lot of weight gain over a lifetime.
Exactly that. There's an astonishing statistic that if you eat 7 calories more a day, it translates to something like 10 kilos extra when you reach middle age. It's only a very subtle imbalance that's needed to end up with quite a profound and health affecting weight gain.
Implications for clinical interventions then off the back of this?
Well, our particular niche bit of biology to do with DEN-D1B is informative because it's acting in a pathway that is already a target of anti-obesity drugs, not the most common kind of ones that have been in the news recently, but others. And so that's important. We need to understand the nuances of these mechanisms in order to be able to develop drugs to target them. I think the kind of wider implications actually from our study come from the fact that dogs are such a relatable model, and the fact that we had this ability to quantify, to put a number on how at-risk dogs were, to show that that was acting via altering their appetite. And then we went on to look at the impact that owner management had on that. And what we showed is that if you're a low-risk dog, actually, it doesn't matter that much what your owners do. You'll probably stay about a healthy weight, maybe a little bit overweight. Whereas in our... That was the puppy in the background rattling things.
This is what happens, isn't it?
Exactly.
Never do experiments or work with children and animals.
There you go. And I'm completely susceptible to being distracted by cute puppies. Who'd have thought it that a veterinary researcher was like that? Yeah, the low-risk dogs tended not to get overweight, whereas our high genetic risk dogs were really dependent on their owners to exert lots of control over what they were eating and make sure they were really active. So if you were a high-risk dog with a completely on-it owner who absolutely regulated your food and gave you lots of activity, you could be perfectly slim. The problem comes that if you're a bit relaxed about the management, those dogs will pile on the pounds.
Eleanor Raffan from Cambridge there. Lastly, as we hurtle into the festive season, why not give Gabir a go? Now, this is an alcohol-free beer that nevertheless still gives you a buzz, but without the headache the next morning. And not only will you be hangover-free, but your body won't be inflamed, nor is there a risk of addiction. In October, I spoke to the creator of Gabir, who's neuroscientist David Nutt, to get a taste of what all this is about.
So Gabir is a new functional beer, but the name is a pun on beer and GABA. And GABA is the molecule in the brain which alcohol works on to keep you relaxed and calm. Primary effect of alcohol to improve sociability and conviviality is by enhancing GABA. So our company is called GABA Labs, and we are working with various molecules, largely botanical, but also synthetic, to mimic the effects of alcohol. And currently, we are selling Gabirs, which are alcohol-free beers that have the flavor of classic beer, but which have the functionality induced by herbs, which promote GABA in the brain, and that helps the people who drink them get the effects that they want from alcohol, and which they do not get from alcohol-free beers.
Are they alcohol-free? Because some people will argue, well, you need a bit of alcohol there because it does contribute to the flavour and the volatility of a beverage.
We have gone for the very, very low alcohol level, which is still designated as alcohol-free, because we want the best taste. So our beer and our stout are brewed in the classic way, and then the alcohol is taken out. So there's just a small residual of alcohol. And as you point out, that probably also contributes a little to the mouthfeel and the taste and the volatility. So you get maybe a little bit of that first impact on your senses before the botanical ingredients go into your stomach and then into your blood and then into your brain.
When are the botanicals added then? Do they go in after you've made the beverage and taken the alcohol out?
Gabir is actually a real Arabic word. It means learning. It is a botanical liquid which we have developed over many years to mimic the effects of alcohol. We've put it into other drinks. Some of you may know of our first drink, which was called Sentia, which is a series of interestingly flavored spirits. Then we've moved on to take out of those key ingredients that produce the functionality, and we've called them Gabir. We've also been able to put them into an alcohol-free whiskey, which is called cask. So these botanical ingredients give people the functionality that they're looking for, but without the harms of alcohol.
What was the insight for finding these things? Did you rationally search for these herbs, or did you hear a whisper on the grapevine from traditional folk knowledge that this could do this? How did you discover this particular combo in the first place?
We searched biochemistry databases, botanical databases, to find herbs that contained compounds which were known to enhance GABA. And then the second step was to select ones that are already in the food chain so that they're approved as food or food supplements. So we could then put them into drinks and market them as botanical beverages with a functional effect.
Is this completely legal, as in unregulated, and you don't have to worry about someone coming along and saying, well, you're drugging the population if they drink this and this is now a Class A drug or something? How do you get around that?
Well, these are botanical herbs. You can buy them and eat them. All we do is put them into a liquid so you can drink them.
And there's no long-term health consequences, there's no hangover effect, there's no onward impact, as it were. They just have a here and now effect on the GABA system in your brain.
People haven't been drinking our drinks for tens of years, but the fact that these herbs are approved as foods means that the authorities have decided that they are safe if they're used on a regular basis as foods. So our supposition is that they will have similarly little negative effects in the long term if they're used in drinks. And one of the real benefits of these that people often comment on is the fact that you do not get hangovers because they do not produce the inflammation in the body that alcohol does. And also you don't get very drunk and you don't get very angry, and they don't seem to cause addiction because they don't target the other chemicals in the brain that alcohol works on to give its unwanted effects. We can minimize the harms of drinking.
How quickly does it start to kick in? How long does the effect last for? How intense is the effect?
It doesn't matter how much of this you drink, you will not get blind drunk, you will not have blackouts, and you will not have hangovers, because we have made these botanical mixtures to produce what we call a plateau effect. So there's a maximum they can achieve, and however much more you drink, you won't exceed that. The effect will last longer, but you won't get a greater effect. The drinks are constructed so that the effects come on within 10 to 15 minutes, and we do that using other herbs. So as well as the herbs which give you the effect, we also put in other herbs which accelerate the uptake of the active ingredients through the gut and into the brain. The effects are designed to be relatively short-lived, so generally they last for less than an hour.
There's a claim that if you drink fizzy beverages like champagne, it makes your stomach empty faster, so you get drunk quicker.
It is true. No, fizzy drinks accelerate the uptake of alcohol, which is why it's quite dangerous to drink fizzy drinks at the start of a drinking bout, because you get drunk faster, so you start to lose control sooner. Actually, a lot of our customers use fizzy mixers with Essentias. In fact, with the cask, the whiskey alternative, I like to drink cask with a caffeine-free cola.
What's the market looking like, and how big could this get? And can you protect this? As in, what's to stop me taking your drinks, shoving them in some very good analytical equipment, working out what your molecules are, and then just ripping them off?
I mean, you could mix the herbs together as we've done, but you don't know the exact amounts. That's a trade secret. And why would you? Much better to do what companies are doing now, which is coming to us and saying, please, can we put your active botanical ingredients, your gabirs, into our drinks? We've been approached by 2 global drinks companies to see if they can improve their alcohol-free beers by adding in our ingredients. So yes, you could try to do it. It's very difficult. It's taken us 6 years. So it would take other companies quite a long time. And by the time they've got to where we are, we're likely to have moved on quite a lot further in terms of having more specific, more powerful ingredients, and ones with even less colour, so we can put them into vodkas and Bacardis, etc.
That was David Nutt, so something to contemplate if you are considering a booze-free beginning to 2026. That's us done for 2025, but before I go, huge thanks to all of you who support this programme by listening to us, by sending in reviews and feedback, and also generously donating to the show to help us to cover our running costs. And if you'd like to put something into our Christmas stockings this year, please head over to nakedscientist.com/donate. We'll be extremely grateful. We've made it really safe and simple. Lastly, thank you very much to our producers Rhys James and Rachel Ralph for their sterling work. The Naked Scientists is sponsored by Rolls-Royce. I'm Chris Smith. Have a wonderful Christmas and a very happy and healthy New Year. Goodbye.